Welcome to this lecture on the fundamentals of genetics. Today, we are going to explore two fascinating phenomena that challenge the classic rules of inheritance: incomplete dominance and codominance. If you have ever wondered why a red and white flower can produce pink offspring, or how human blood types are inherited, you are in the right place. Let us dive in and uncover the beautiful complexity of genetic expression. To begin our journey, let us look at incomplete dominance, often called the blending effect. Why does this matter? Because it explains why not all genetic traits fit neatly into simple either-or categories. Imagine mixing red and white paint to get pink. In genetics, when one trait is not strong enough to completely hide the other, the offspring ends up with a physical appearance that is a perfect blend of both parents. However, it is crucial to understand that the underlying genes themselves are not permanently blended. They remain distinct and can separate again cleanly in future generations. Now that we understand the concept of blending, let us look at the mathematical formula that predicts this behavior in the second generation. This equation comes from the random assortment of alleles during reproduction. The variable p stands for the probability distribution of the offspring's genotypes. The symbol C superscript R represents the allele for red color, while C superscript W represents the allele for white color. The fractions 1/4 and 1/2 indicate the expected mathematical frequency of each genetic combination. If the total number of offspring increases, the actual observed numbers will closely approach these exact mathematical fractions. Before we move on to the second generation, let us see this formula in action with a concrete example. Suppose a botanist crosses a pure-breeding red snapdragon plant with a pure-breeding white snapdragon plant. We are given the homozygous parents and asked to predict the exact physical appearance of their immediate offspring. We assign C superscript R for red and C superscript W for white. The pure red parent can only give the red allele, and the pure white parent can only give the white allele. Therefore, every single offspring receives one of each, resulting in a completely pink flower. So, we have successfully found the first generation. This brings us to an important question. What happens if two of those first-generation pink offspring are crossed with each other? We are setting out to prove that the original color genes are not permanently destroyed by the blending effect. We take two pink flowers, both with the heterozygous genotype C superscript R, C superscript W. During reproduction, each parent has a 50% chance of passing on the red allele and a 50% chance of passing on the white allele. Let us see how these probabilities combine. Continuing our derivation, we multiply the probabilities of these independent events. This random assortment results in 1 out of 4 offspring being pure red, 2 out of 4 being pink, and 1 out of 4 being pure white. This perfectly matches the mathematical equation we saw earlier, giving us a 1:2:1 ratio. This final result clearly proves that the original red and white traits were never destroyed or permanently altered. They simply hid within the pink generation and reappeared perfectly intact in the next generation. Now that we have proven the math, let us visualize this beautiful mechanism. On screen, you are looking at the complete genetic cross for snapdragons. At the very top, we have our pure red and pure white parents. Following the arrows down, you see the first generation, which consists entirely of blended pink flowers. Below that is the Punnett square for the second generation. The axes show the alleles from the pink parents, and the inner grid reveals the 1 to 2 to 1 ratio of red, pink, and white flowers. This diagram perfectly illustrates how alleles compromise without losing their underlying genetic code. Having explored incomplete dominance, let us shift our focus to a different scenario called codominance. Why is this important? Because in human medicine, particularly blood transfusions, understanding this is literally a matter of life and death. Imagine a red cow and a white bull having a calf. The calf is not pink. Instead, it has distinct patches of pure red and pure white hair. In codominance, the traits refuse to mix. Both alleles are fully and simultaneously expressed in the offspring. Neither allele is masked or diluted. The cellular machinery reads both genetic instructions and builds both proteins independently, sharing the spotlight. Just as we did with snapdragons, let us look at the mathematical formula for codominance, specifically for human blood types. Conceptually, this formula arises from the exact same random assortment of alleles, but the physical result is entirely different. Here, P again stands for the probability of the offspring's genotypes. The symbol I superscript A represents the allele that produces the A antigen on red blood cells, while I superscript B represents the allele that produces the B antigen. Because both are codominant, the middle term represents individuals who have both A and B antigens perfectly expressed, giving them the unique AB blood type. Before we look at the next generation, let us apply this formula to a real-world medical example. We are given a mother with pure type A blood and a father with pure type B blood. We need to determine the exact blood type of their child. The mother is homozygous, meaning she can only contribute the I superscript A allele. The father is also homozygous and can only contribute the I superscript B allele. Therefore, the child must inherit exactly one of each. Because these alleles are codominant, the child's red blood cells will manufacture both antigens simultaneously, resulting in a type AB blood phenotype. Now that we have found the child's blood type, we must explain what happens if that child eventually grows up and has a baby with another person who has the exact same type AB blood. We are setting up a mathematical cross to demonstrate how codominant traits are passed down through multiple generations. We take two parents, both with the heterozygous genotype I superscript A, I superscript B. Just like before, their alleles will separate randomly during reproduction. Let us calculate the expected distribution of blood types for their children in the next step. Completing our derivation, we see the mathematical probabilities unfold. There is a 25% chance the child gets two A alleles, a 50% chance the child gets one of each, and a 25% chance the child gets two B alleles. This final answer demonstrates that codominant alleles maintain their distinct identities without blending. Notice that they follow the exact same 1 to 2 to 1 mathematical ratio as incomplete dominance, even though the physical result on the red blood cells is completely different. Both original traits are preserved perfectly. To truly grasp this concept, let us look at this diagram of red blood cells. On the left, you see a type A red blood cell, covered exclusively in A antigens, represented by specific markers. In the middle is a type B cell, covered entirely in B antigens. But look at the type AB cell on the right. It does not have a blended intermediate antigen. Instead, it proudly displays both the A antigens and the B antigens at the exact same time. This visual perfectly captures the essence of codominance, where both genetic instructions are fully and independently expressed. We have covered a lot of ground today, so let us summarize the most important ideas. First, incomplete dominance is like mixing paint. It creates a blended, intermediate physical appearance where neither allele completely wins. Second, co-dominance is like a tie game where both teams get a trophy. Both original traits are fully and simultaneously expressed without any blending. Finally, despite their different physical results, both mechanisms follow the exact same 1:2:1 mathematical ratio in the second generation, proving that our underlying genetic code remains perfectly intact. You now have a solid understanding of these fundamental genetic principles.